Hi, I'm Jay Phelan. Let's talk about some biology. What I want to do today is to look at the ways that we can use linkage data to build chromosome maps. So going from recombination frequencies to an actual physical structure of a chromosome. Let's start with a genotype that is something we've been looking at a lot when we look at studies of linkage. If we have an individual that's heterozygous for each of two different genes, so we'll just say big A, big B, little b. We know how that we could obtain that already by getting something that's true breeding for each of those two genes. When we have the double heterozygote, though, we're going to do the cross that T.H. Morgan first pioneered, where we test cross it. So we're crossing it with an individual that's homozygous recessive for each of the two genes. The reason we do this is that when we make a cross, we're going to get 4 classes of offspring. And for each offspring, remember that the test crosser is going to provide a little a and a little b. That's all it has. So in every one of them, I've indicated the little a, little b that came from the test crosser. Now we look at the other allele that the individual carries for each of the two genes, and we know that that is what the genotype was for the gamete that individual produced. And if we know what gametes it's producing, when we see a deviation from 25%, 25%, 25%, 25%, then we know that they are linked. So we look in this case, out of 1,000 offspring, most of them are big A, big B, or little b. And then we get some that are big A, little b, or little a, big B, which means that those weren't stuck together on the initial individual, but they're produced because of recombination. So what's going on here? We would say that from these gametes that are produced, we can tell that the two genes are linked, and we can even tell something about how far apart they are. We look at the crossover gametes, big A, little b, and little a, big B. And what do we get from those? We get about 10% of the total gametes instead of the 50 that we would get if they were not linked. So we know that they are 10 map units apart. So we've already covered this, but it's just a refresher that when you look at the crossover gametes, the proportion of those is going to tell us how far apart they are. So that was a previous take-home message that we've got. Map distance is the total percentage of gametes that result from recombination. So now let's take a look at this. What if we look at not two genes, but we're interested in notch wings, cross-veinless wings, and raspberry eyes? Now, notch wings is a mutant form of Drosophila wings we see, and you'll see that it's manifest in a variety of ways, but you have little chunks that never develop in the wing. So it's pretty easy to see physically and coded for by a recessive single gene trait. Then we've got cross-veinless wing. When you look at a fruit fly wing, they actually have their veins in the same place all the time. So the wild type on the top, those are where the veins are typically going to be. But in cross-veinless, you notice right here, there's no vein. So again, it's something that's easy to see in the phenotype. And then one final one, raspberry eyes. Typically we have red eyes in fruit flies, but occasionally you'll get something that is a slight variant on that. For these 3 genes, if we do our standard create a double heterozygote, do a test cross, figure out what percentage of recombinant gametes are formed, we can determine that the distance between notch wings and cross-veinless is 10.7 map units. You should be able to figure out what the cross would look like, what the gametes would look like. Then if we do a separate experiment and we look at the distance between notch wings and raspberry eyes, we see 29.4 map units. So we know that we can map out from notch wings to cross-veinless, 10.7. And then from notch wing to raspberry, 29.4. I've put them on a map here with N in the middle, and we're going in opposite directions to get to each. Chromosome is a linear structure. But this isn't the only map we could draw. Think about it. We go 10.7 to cross-veinless. We don't have to go that way to raspberry eyes. Maybe it's the other way. It could be in either position, but we want to know because there is a chromosome and the genes do have a physical location. Here's what our other possible map would look like. Same distances between notch wing and the two other genes, but in one case we put it on one side, one case we put it on the other side. But here's the thing that's interesting here, that the difference between these two maps is this. If we do a third experiment, we look for the proportion of crossover gametes produced when we compare raspberry eyes to cross-veinless wings. If the map distance we get is 40.1, they're really, really far apart, that tells us that map 1 is correct, that distance has to be that. On the other hand, if we do that, calculate those map distances and we get just 18.7, then we know it was notch, cross-veinless, and then raspberry, so that there's a smaller distance here. And you could do this here for these 3 genes. We figure out which one it is. This was done by one of T.H. Morgan's students, a guy named Alfred Sturtevant, and he did it when he was an undergrad. He was 19 years old when he first started, published when he was 21, showing that you could get these genetic maps. On the right side is much later in his career, but there he is on the left as a youngster. What he did was treat it like a puzzle. We did this experiment, got this distance. Same gene with another one, we got another distance. Now, how do we figure out which relative position they're on? And he did this methodically over and over, looking at the data that they had already obtained. Scoot bristles, then notch wings, then cross-veinless has to be here, then raspberry eyes. Those are notch, cross-veinless, and raspberry, we already saw. Miniature wings, forked bristles. So for any pair where you have 2 crosses and you know the distance from a gene to one and then a distance to the other, by comparing the 2 other ones, where are they? We could figure out an entire chromosome map. So he did this over and over and over again until he was able to place every gene on the various chromosomes that they were linked with. From the data that we look at, you should be able to figure out what the maximum recombination frequency is. And what does that mean even? Think about that when they're very, very close together, the space allowing for recombination is small. So the frequency that's going to happen is very low. As you get a larger and larger and larger distance, now you have more space in the DNA that you can have a crossover occur. But what's the maximum going to be? And why might the maximum distance be less than the actual genetic distance? We see forked bristles is, 56.7 map units away from scoot bristles. That's actually going to be a larger physical difference than the recombination frequency. So think about why that would be. So our take-home message here is that when we measure recombinant frequencies, that tells us the map distance. But when we have a lot of these together that have some overlap of what they're comparing, then that allows us to map out the distance between genes and to figure out the relative locations until we can get a full map of where everything is. And that's it. That's how we use linkage data to build chromosome maps.